Okay, fantastic. Thank you again for the introduction, and thank you to the Nevada Native Plant Society for funding my research and making this possible. And also thank you for allowing me to tell you guys all about buckwheats because I love them so much. Um, and today I'm really excited to talk about buckwheats in Nevada and also a bit of what I'm working on today. And I wanted to preface that, of course, I don't know everything, and maybe you guys know some things I don't know as I go through my talk. So if you guys feel inclined to chime in or ask a question, please feel free to interrupt whenever. It's welcome. Okay. To start off my talk, before I dive into everything, I thought it'd be fun to go over the evolutionary slash taxonomical background of Eriogonum. So like, what does it mean to be a wild buckwheat in the context of all living things? And the way that I thought to explain this was by going through what is called the taxonomic hierarchy. And the taxonomic hierarchy is essentially a way for biologists to organize all living things. And the categories, as I visualize here in this chart, set out very broad. And then as you go down these categories or ranks, the more specific you get. So the first rank that we have is kingdom, and this is where we have our kingdom of plants, of fungi, of animals, and others that I've not pictured include like of archaea, for example. But given that we are in the Nevada Native Plant Society, the kingdom that we'll be staying in today is the plants. Next down, we have the phylum, and within the plant world, some of the main phyla are of the bryophytes at the top. So those are our non-vascular plants that include our mosses, hornworts, and liverworts. And another phylum is our cone-bearing plants or gymnosperms, like our pine trees. And the other phylum is of the flower-bearing plants or angiosperms. Eriogonum is a flowering plant, so that is a phylum that we are in today. Now, within the flowering plants, there are two main classes. The top photo I have is of an iris representing the monocotyledons or monocots. And these are plants that are characterized by having their flower parts in three. So you have three petals, for example. And their roots are fibrous. So if you ever had to do weeding and pulled out a grass, that's a monocot with fibrous roots. And their leaves have parallel veins. Contrast that with the lower picture I have is of a phlox, representing the dicotyledons or dicots. These guys have their floral parts in fours or fives, like we see here. Their roots tend to be taproots. If you think of a carrot, that is a taproot, part of the dicotyledons. And their leaves have palmate veination. And Eriogonum, I realize this is unintentionally a bit of a trick question. Eriogonum has six, or like their flower parts in threes, but they are in fact dicots. But I'll talk about that a little bit more later. As for order, that is the next rank that plants are organized in. And there are way too many orders to show as a flow chart. So I'm just going to tell us all that we are in the order Caryophyllales. And even if we haven't heard of the name Caryophyllales before, I feel like we're going to be familiar with some of the plants in this group. So the way that orders are put together is by being a monophyletic group. So monophyly or monophyletic is a word that I will be using a lot throughout this talk, so I thought I'd introduce it here. And monophyletic means when all of the members of a group have or share a common ancestor. So the order Caryophyllales, all the members of this group share a common ancestor. And even though we know that, there's not really a clear characteristic that ties this whole group together. And you'll find that this group is super diverse. We have beets, for example, cacti are in this order, our buckwheats, of course, in addition to carnivorous plants and bougainvilleas. Now, even though Caryophyllales as a whole doesn't have one, like a clear uniting characteristic to hold it all together, there are two main groups that kind of have uniting characteristics. And one of the groups are the caryophyllates, and they are represented by having the presence of betalains, which is a type of pigment, instead of anthocyanins. And a way that I like to remember this is that beets are in this group. So betalains, beets, I don't know which came first, but they're related somehow. So that's where that red pigmentation comes from. And the other group is the polygonids, which includes a carnivorous plants. But aside from that, there's not really a clear uniting characteristic. But fun enough, our wild buckwheats are in this group. So that's where we are. The next rank below order is family. And again, there's too many families to show here. So I'm just telling us all that we are in the family Polygonaceae. So let's talk a little bit more about that. So Polygonaceae is also broken into two subfamilies, but something else to note is that Polygonaceae is known for having these swollen nodes or like a bit of like and like outer like inflation where the leaves are inserted, kind of like what we have in this picture of Persicaria. I don't always see that in all of Polygonaceae plants, but I do see it here, so it does show up in this family. And one of the two subfamilies in this family is Polygonoideae, and they are characterized by having this feature called an ochrea. And what an ochrea is, if we're not familiar, is yes? Oh, sorry. I thought someone had a question. Um, what an ochrea is an extension of the leaf base, and it forms a sheath around the stem. And some examples of these are rhubarb, so rheum in the genus rheum, as well as Phagopyrum. Now, if you look at the plant itself, we may or may not recognize it, but Phagopyrum is the genus that is what is used to make the commercialized buckwheat flours. If you heard like buckwheat pancakes and whatnot, that is where buckwheat flour comes from. Oftentimes, when I tell people that I study wild buckwheats, they ask me if I mean like the ones in buckwheat flour, and I would have to reply with, "Well, they are in the same family, but they're just in a different genus." And so this is the genus that you get your buckwheat flour from. Contrast that with the other subfamily is Eriogonoideae, and they're characterized by basically not having an ochlea. And this is, spoiler alert, where Eriogonum falls. And here's a close-up of the California buckwheat. And if you look where the leaf base is, it does not form a sheath around the stem. So that's how you know you're looking at something in the Eriogonoideae subfamily of Polygonaceae. And because we're focusing on this group, I just thought to mention a couple of more features of this subfamily. In addition to not having an ochrea, they have these structures that are called involucres, which are modified leaves that fuse and form a cup-like structure to hold the flowers. So that's what we see up here with this acanthocypis. We have the fused leaves with these really fun projections called awns, and the flowers are nested in there, kind of like a little vase or floral bouquet. And I mentioned a little earlier that buckwheats, even though being dicots, have the floral parts in threes. So that's what we have here. They actually have 6 tepals. If we are not familiar with the word tepals, that's basically a word that botanists use for when we cannot tell visually apart the sepals, or like what's usually the green part versus the petals. So in that case, we just use the word tepal. And some examples of some genera in this subfamily include, of course, our Eriogonum, but also our Coryzanthe, the spine flowers, Nemacollis, which are found on sand dunes, Acanthocypis, and Oxytheca. And now we have made it all the way down the taxonomic hierarchy to our very own genus, Eriogonum. And now, how do we tell we found an Eriogonum? So here's some like the top main features that I like to categorize Eriogonum in, more like visually. And one is that their leaves are basal or like at the bottom, or they could have both basal and cauline leaves. And cauline leaves are basically leaves that are further up on the flowering stem. And in Eriogonum, their involucres or their cup-like structures, they don't have awns. So that's how you tell them apart from other genera. And as you mentioned earlier, they have 6 tepals, but also they have 9 stamens. So those are the pollen-bearing structures, or what's pink in this picture. And in general for Eriogonum, it is further broken down into 8 subgenera. 4 of them can be found in Nevada, so we'll be talking about that today. As a whole, they have a Western North American distribution, with a lot of them in the Western United States. There are over 250 species total, and 80 of those are represented in Nevada. Now, if we bump that number up to, if you count varieties, that number bumps up to 120 species and varieties in Nevada. So that's pretty impressive. There is an incredible amount of morphological diversity that we'll get to explore today. And in parentheses, I note that Eriogonum is not monophyletic. So in contrast to when I was saying that the order Caryophyllales is monophyletic, meaning all the members have the same common ancestor, Eriogonum does not have that. And that's something that I'll be touching on as I go through this talk. But in the meantime, I thought it would be nice to take a little look at the wonderful morphological diversity in Eriogonum. As far as habit, or like how their growth structure is, they could be herbs like you have with Eriogonum zyonis, or you could have these shrubs like the California buckwheat and Eriogonum giganteum. You can have these little tiny mat formers that grow on like limestone rock, like Eriogonum gilmanii. And you can even have these teeny tiny buckwheats that are like this tall. In addition to these different morphological structures, they also vary in color. You can have flowers that are white and green to white and pink. You can have yellow flowers with tinges of red and pink, and they support a variety of native pollinators. Okay, so this is a part where I'd like you guys to somehow participate if you are so inclined. But because we learned a bit more about what is Eriogonum and how to recognize one, I thought you'd play a little game inspired from when I would go out hiking. And you can tell me if you relate to this, but if you're like hiking and you see from a distance a plant up a canyon wall or a slope and you swear it's an Eriogonum, but then you go up close and it's not. Usually for me, it ends up being some sort of sunflower. So I kind of took pictures of plants that kind of look like buckwheats, and I wanted us to choose which plant we thought was Eriogonum. So first up, I have this, and I'm trying to figure out how to do this over Zoom. But I guess if you want to unmute, you can say if you think it's the one on the left or the right. We can call the left A and the right B. You can vote in the chat, or do some, maybe do a thumbs up for left and heart for right. It might be a little chaotic, but we'll see how this works. But in the meantime, which one do you think is Eriogonum? You have to just wait for me to see the chat. Okay, so you got one vote for right. Any disputes? Okay, we got 2 for right. Okay, you got many people saying it's on the right. Okay. Oh, great. Cool. Las Vegas says it's the right. Alrighty. If we're ready, I'm gonna reveal which one is Eriogonum. Ready or not, here we go. Let's see. You guys were right. It was the one on the right. The one on the right is Eriogonum aballifolium, or the cushion buckwheat, and the one on the left is actually a Petrophyton in the rose family. Okay, we're gonna do one more of these. Which one is Eriogonum? Okay, so I need to write again. You guys are good. Awesome. Yeah, you guys are really good. You guys are buckwheat experts at this point. Should take you out in the field with me. But yeah, the one on the left is actually Sambucus nigra, or the elderflower. Cool. Alrighty, so thank you for humoring me. I hope that was at least a little fun. I'm having too much fun over here myself. But now that we learned this whole background of where Eriogonum falls in the tree of life, and we got to play a little game to test our Eriogonum eyes, let's get into some of the Eriogonum of Nevada. Because there's over 120 species and varieties, I won't be going over all of them, but the way I thought I'd present this is by going through the different subgenera. that are found in Nevada and go from there. So I wanted to start off with the subgenus Oligogonum. In this subgenus, there are 35 species in total. 8 of them can be found in Nevada, and 1 is actually endemic to the state. And they are perennials, so plants that live for more than 1 year. And another physical characteristic they have in common is that they have what is called a floral stipe. And I tried to show that in a photo here in the sulfur buckwheat or Eriogonum undulatum. But a floral stipe is a structure. Well, it's probably better explained if I say what when you don't have a floral stipe. So if you just imagine a flower like a tulip, and then my arm is like the stalk or the pedicel. So there's like a joint right here where the flower attaches to the pedicel. And that's like a normal flower that does not have a floral stipe. But if you do have a floral stipe, the flower itself narrows into a tube before it attaches to the stalk. So when you have that narrowing of the floral tube before the pedicel attachment, that's when you have the floral stipe. So if you're ever out there wondering if you have an oligogonum, that's a feature that you can look for. Here's some members of this subgenus. The one that is endemic to the state of Nevada is a sunflower flat buckwheat pictured here in the middle. This is Eriogonum douglasii, variety Elcoense. And some other highlights from this subgenus include Eriogonum cespitosum, Eriogonum labii, and Eriogonum heracleoides. And a little bit of a fun fact for the middle one, Eriogonum labii was one of my bucket list buckwheats because I really wanted to see if it smelled as bad as people said it did. So when I finally got to see it, I did smell it. It wasn't as strong as I thought it was going to be, but it is not a great smell. So let me know if you have your opinions on this buckwheat. The next subgenus is Eriogonum, and contrasting that with Oligogonum, these guys are all annual, so species that live for just 1 year. There are 28 species, and 8 of them are found in Nevada. None of them, as far as I know, are endemic to the state, but if anyone knows otherwise, please let me know. And another feature that ties this group together is that their involucres, or like their fused leaves that form this cup-like structure, they are at an angle and they are oppressed to the branches. So in a way, they're like fused up against it. And that's showed here in Eriogonum palmarianum in the center photo. And another representative from this group is a bird's nest buckwheat. And this is one of the cutest buckwheats I've seen. You can kind of see right now where when it's green and in flower, it kind of has like this bird nest shape, but it really looks like a bird's nest when it dries and it turns like this golden red color. Here are some other representatives from this subgenus, including Eriogonum bailii and Viminaeum. Something I've learned about annual buckwheats is that they're very hard to photograph, especially if you're trying to do a whole plant photo. So oftentimes the best photos are when you can get them up close. The next subgenus we're going over today is the subgenus Eucycla. It is the largest subgenus in Eriogonum with over 100 species in total, 36 of them found in Nevada and 12 of them endemic to the state. They are perennial herbs and shrubs, but aside from that, there's not really some clear uniting characteristic. But some representatives from this group include the California buckwheat, as well as a crispleaf buckwheat. And as for one of the endemics that I wanted to highlight is Eriogonum teamii, or the Teams buckwheat. And I wanted to highlight this especially because it is an Eriogonum of conservation concern. It is actually listed under the Endangered Species Act as endemic, as endangered. And it is endemic to these outcrops that have these soils that are rich in lithium and in boron, and it is under threat for mining and whatnot. So I'm really grateful that it is having this federal protection, and I hope that it's doing well. If anyone has updates, let me know. But I have a sticker that shows I'm on Team Buckwheat. So yeah, hope it's doing well. And here are some other endemic buckwheats in this subgenus, including the West Humboldt buckwheat, the Churchill Narrows buckwheat, Holmgren's, Lewis's, and the Cloquet buckwheat. Last but not least, the fourth subgenus represented in in Nevada is a subgenus Ganysma, which by no biases at all is my favorite subgenus. It is the second largest one with 62 species total, 27 of them found in the state of Nevada, 5 of them endemic to this state. They are mostly annual species with a few perennials. And the way that you can tell these guys apart from, let's say, subgenus Eriogonum is that their involucres are not oppressed to the stems, they are free hanging. And here are some representatives from this group. We have the pagoda buckwheat, which is very charismatic, and I actually have some dried ones here that Steve Bryant gave me. They dry really well. They're very charismatic. And another buckwheat in this group that is endemic to Nevada is Eriogonum cancinum, or Darren buckwheat. This one is a really fun one, and the photo here is by Jeanette Perry. I was really lucky that I got to see it in person because not only is it endemic to Nye County, Nevada, but the only known occurrences are within the Nevada nuclear test site. And I was super lucky that I got permission to go collect there and see this plant in person, see the habitat, make some collections. But the only catch is that I couldn't use my phone or take photos. So the botanist that accompanied us, Jeanette Perry, she was kind enough to take photos on a digital camera. And once they got declassified by the government, she sent them to me. So these are declassified photos from the Nevada test site of one of my study plants. And here are two other endemic species to the state of Nevada. We have the Death Wild Buckwheat, Eriogonum nutans variety glabratum, and that photo is from its type locality, or the place that it was described, which is this gravel lot, and it is a taxon of conservation concern, especially for me, because the past times I've gone there, there have been like construction vehicles nearby, and the plants are doing really well where they are, but I am concerned about their well-being with this construction and development around it. So I'm hoping to get on top of that for getting some protection, as well as Eriogonum lemoni, or the lemon buckwheat. And I wanted to highlight some conservation implications. So far in my talk, I have highlighted a little bit about conservation in this genus. There are 18 endemic species and varieties. I highlighted a couple of them as you were going through, but that is something I want to point out, that there are quite a few endemic species and varieties in the state of Nevada. And 2 of these are listed under the Endangered Species Act as endangered, and 4 are listed as Nevada state protected species. And something I also noted at the bottom is that that a lot of these species and varieties are rare and restricted because of specialized adaptation in addition to threats. But another thing that restricts them, and the reason why they're so rare, is that they are specially adapted to certain habitats. I mentioned Eriogonum temei, or the Teams buckwheat, that is endemic to these soils high in lithium and boron. And the other example I have pictured here is Eriogonum ovalifolium, variety williamsensi, or the Steamboat Springs wild buckwheat. This buckwheat is also listed under the Endangered Species Act, and it is endemic to this one locality at the Steamboat Springs area, where it is growing on these outcrops that are silica deposited soils. So it is very much reliant on like this hot springs area and is in need of protection. And of the 4 Nevada state protected species, one of them is in my study group as well, Eriogonum vestigium, or the sticky buckwheat. And so with that, I wanted to kind of like shift gears into talking about my research in the subgenus Ganysma and like the conservation implications of that. So yeah, let's get started. As I mentioned earlier, there are 62 species, but if you count varieties, there are 74 species plus varieties in subgenus Ganysma. Most of these are annuals, as mentioned before, but there are a couple perennials, and they have a mostly desert distribution. And that's one of the reasons that I studied them, is that because they're annual and they live in the desert, they are understudied, which is something I found is hard to study, because not only is it really hot and dry out there, but being annuals, they may or may not come up depending on the year and the rains. But another reason I chose to study this group, besides being fascinated by them and their adaptation to the desert, is one more reason, and that's the Eriogonum deflexum group. So I actually began my study by focusing on this group of 16 species and varieties within what is called subgenus Ganysma, that have these upside down or deflexed flowers and involucres. So that's kind of pictured here. And they may or may not have these structures that are called peduncles, and peduncles are these stalks to the involucres. But if they do have peduncles, they're always hanging upside down. And the objectives of my study can be broken down into these four main parts. And in my research, I am uncovering the units of diversity. And what this means is that I'm trying to figure out who's who in terms of the species. And I'm doing this by sampling all of the members of what is currently called the subgenus Ganysma. And with this, I'm testing the current classification. And what that means is that I'm trying to see if the current names are holding up. And this is because a lot of times plants and other organisms are classified based on just morphology or how things look. But recent molecular studies, genetic studies, are showing that when you sequence their DNA, that there's more diversity than is being counted for when you're just looking at morphology. So I'm using what is called phylogenetics, or looking at relationships based on genetics to see if these hold up. And with this information, I am informing conservation because by figuring out who is who and identifying vulnerable populations, we can make these conservation recommendations based on their critical habitat, and we can figure out how rare they are. And the fourth thing I'm doing is studying phytochemistry in a phylogenetic context. And phytochemistry is essentially what chemicals are found in the plants. And I'm doing this for a couple of reasons. One, I'm curious to see if there is a genetic signal to these different compounds. And I'm also curious to see what compounds these plants have because they are adapted to these extreme environments, like that picture of Eriogonum rexfortii or the pagoda buckwheat. That picture was taken when it was like 119 degrees outside, but that plant looked quite happy being where it was. And next, I wanted to get into my research progress, which includes going over the fieldwork I've done, what lab work, and also I'll be talking about some of my preliminary results. Starting with fieldwork, I am very lucky in that my fieldwork is essentially road tripping across the Western United States in the search of these lovely plants. It has been really cool and super informative to be able to see these plants in person in their native habitats. And I wanted to highlight some of like my collection process. One of the things that I do is I make these press vouchers for herbarium. For herbarium specimens, and I collect leaf material for both molecular and chemical analyses. And in that bottle, I also have this liquid preservative that I put leaves, stems, and flower pieces in for anatomical analyses. And in addition to these physical collections, I also take very detailed notes on where I collected the plant, what the habitat's like, any of these physical features of the plants, like their color, for example. And I take photos of the habitat, the whole plant, as well as close-ups. Fieldwork has also been a really great opportunity for me to teach individuals how to do botanical collections. So I've been able to take individuals who are curious about how botany fieldwork is and bring them out in the field with me. And these included undergraduate students or post-bac students. So that's been a lot of fun. And I've also gotten to collaborate with other fellow botanists. And to give an idea of what my field work is like, here's like a little map showing my collection points across the Western United States. So I really have been over the whole Western United States, and that's been truly a pleasure and honor. And back in the lab, I process the samples to extract their DNA, and then later I'll be talking about extracting the chemicals. But for extracting the DNA, the process includes hand grinding the leaves with the mortar and pestle with liquid nitrogen and mixing in these chemical compounds that break down the cell walls and disintegrate other cell parts except for the DNA. And after doing some rinses with ethanol, then we end up with hopefully a pure DNA sample that can be quality checked using gel electrophoresis. And once you have a batch of about 96 samples, you can then prepare them for sequencing. And when you prepare them for sequencing, that's called a library. So that's a word I will be using. And so that's how I prepare them. I have all 96 of my samples, and we prepare them for sequencing with, I think, three main steps. So like one, I should preface that the type of sequencing that I'm doing is called dd-RADseq. And this is a type of sequencing that you have, if you imagine a whole plant genome, it is both financially and computationally very expensive to sequence an entire genome of a plant. So what dd-RADseq does is that you have your genome and then you have these enzymes that cut up the genome in different spots, and then it sequences those chunks. And that way you are getting kind of like a snapshot view of the genes across the genome without having to sequence the entire thing. And so this also works really well for plant groups that don't have a lot of prior genetic knowledge, and Eriogonum is one of those. So once you cut up the DNA pieces, and then the next thing you do is that you glue on these tiny barcode tags, so a unique tag for each of your samples, and then you do PCR, which is a fancy way of saying that you're making a bunch of copies of DNA. And then after you make a bunch of copies, you hope that the barcodes attach to the samples and you put them all into one tube. And then they get sent off to the University of California, Riverside to be sequenced. Cool. And once they get sequenced, so I rinse and repeat, and I do that about 6 times. And then you get back these raw DNA sequence files, so like just A, T, C's, and G's, and then you do some bioinformatic coding to make what is basically what is called a phylogenetic tree, which is essentially a family tree made based on, in my case, genetic sequences. Now, we're getting into my preliminary results, and if you're anything like me, sometimes looking at results or data can be quite overwhelming. So before we get into it, I wanted to give us, hopefully, a less overwhelming introduction to phylogenetic trees. So a word that I was using a lot earlier is monophyletic. So I thought I would make a visual for what that looks like in a tree, and also what the other options look like if they are not monophyletic. So on the far left, we have a visual representation of a monophyletic group. And in this case, we're saying that species A, B, and C are all related. And if that shows to be true with genetic sequences, then they all share the most recent common ancestor in red. But if you have what is called a paraphyletic group, you are saying that species A, C, and D, they're all the same thing. But then let's say species B is not meant to be related, like it looks completely different, but then it gets mixed in. So that's when you have a paraphyletic group where you have a common ancestor that has all the species except for maybe a rogue one mixed in. Polyphyletic is when the species that you're hoping would be, or that look alike, do not have the most recent common ancestor. So in that case, like species A and C probably have a lot of features that look the same, but then when you sequence them, they are actually not each other's closest relatives, and that's called a polyphyletic group. Okay, so we're gonna look at my tree next, and it might be a little scary, but it's gonna be okay. We're gonna get through it. Okay, so this is my phylogenetic tree of my preliminary results for my analyses so far. And before I get into what to get out of this tree, I wanted to orient us to what on earth we're even looking at. So aside from this being a tree, some things I wanted to note is that you'll notice that there are sometimes triangles and sometimes just like sticks or lines. And the line represents when I have one sample representing a species or variety. But if I have a triangle, that means I have multiple samples representing that species or variety. And I just didn't want to have too many branches, so I just collapsed them all for simplicity's sake. And another thing I wanted to note is that confidence in how the branches are arranged in this tree is measured by a statistical method called bootstrap analysis. And essentially what that means is that if you have 100% bootstrap support, that means you're very confident in how the branches are arranged in the tree. But once you get below 95%, then we're not so sure. So that is why I indicated in the red dots when we have a bootstrap value of below 95. But aside from that, the bootstrap was relatively high. Most of them were 100, maybe a few were 99, but overall, pretty good support for how everyone is arranged in this tree. Another thing I wanted to note is that most of the members in this tree represent species and varieties from what is currently recognized as subgenus Ganysma, except for these two groups. So I added in some species from subgenus Eriogonum and from subgenus Eucycla to give us context for where these species fall in relative to the other buckwheat species. And it is rooted, this tree is also rooted to Coryzanthus spinosa, which is a relative in the same subfamily. And the last thing I wanted to point out in terms of orienting is that if there's a star, that means it is part of the Eriogonum deflexum group, or those buckwheats with the upside down flowers. Okay, now that we are hopefully more oriented to this tree, I wanted to point out that there are a lot of very interesting things going on in terms of how these guys are arranged in the tree. But in terms of our time today, I am focusing on three main points. And the first one has to do with the other subgenera that I included in this tree. You may notice that subgenus Eucycla species end up right smack dab in the middle between the subgenus Ganysma species. And this brings me to my first point that subgenus Ganysma is not monophyletic. So not all the members share one common ancestor, and they're not all one group together. And this supports a study that was done in 2012 by Elizabeth Kempton, and she sampled she sampled across Eriogonum, but also other genera. And that's where she first noticed that Eriogonum is not monophyletic, and also a few species of subgenus Ganysma were sampled, and those did not end up as monophyletic. So that's how I ended up in this study doing more dense sampling to see if this still holds up, and it does still hold up. Next, you may notice by how the stars are distributed that the Eriogonum deflexum group is also not monophyletic. And furthermore, this brings me to the point that deflex flowers have an interesting evolutionary history. So I have here in the green dots, these are the different points at which species with upside down flowers come up. And with this, I have like two main hypotheses. One is that maybe upside down flowers evolved once, and there were some cases that of reversals where the flowers went right side up again, or deflex flowers evolved 4 times. Either which would be really interesting to see, and I'm hoping to do an analysis to try to find out their evolutionary history. And the last point I wanted to share today is that Eriogonum deflexum itself is not one species. It actually shows up in 2 different areas in the tree. And this is something that I wanted to point out a couple more things. Okay, so I wanted to zoom in on this part of the tree to make it easier for us all to see. But some things I wanted to point out is that Eriogonum deflexum has 3 currently recognized varieties. Has variety deflexum, variety nevadense, and variety baratum. As you can see, they are not showing up together as each other's closest relatives. And the first thing I wanted to point out is this group here with variety baratum and variety nevadense. They are forming what is called a paraphyletic group. So this is something that we looked at before. So taking it back to our model from beforehand, just to give us a visual. So the paraphyletic group is how like we have Eriogonum deflexum variety nevadense, but then you have variety baratum kind of nested in it. I also wanted to highlight that we have Eriogonum deflexum variety rectum up here. And if you may notice, it is not one of the currently recognized varieties that I mentioned earlier. In fact, variety rectum is considered a synonym or considered the same thing as variety deflexum. And for the purpose of my study, I tried to sample all the synonyms from this group to make sure that they all are one group. But in this case, variety rectum is not showing up right next to variety deflexum, and so it might be its own thing. So that's very interesting. And the last thing I wanted to point out was that we have Eriogonum exaltatum also mixed in with variety difflexum. And this is very interesting because Eriogonum exaltatum is not in the difflexum group because its flowers and involugers are very much pointed upright or exalted, as the name suggests. So it is very interesting that it ends up nested with variety difflexum. So to kind of give us a bit of a summary of everything that we just saw, so just to confirm, like we saw that subgenus Ganysma, as it is currently circumscribed, is not one lineage. And the implications of this is that, well, I still need to include in my sequencing, I included the species Eriogonum angulosum, which is the type for subgenus Ganysma. So depending on where that falls in the tree, that group will get to keep the name subgenus Ganysma, and then we'll have to figure out names for the other group. Maybe I can do a poll with you guys to find another name. But have to do some more work to see how that goes. And we saw that difflex flowers have a very interesting evolutionary history. It brings questions about whether there's adaptation advantages to having upside down flowers. And we also see that the Eriogonum difflexum varieties need revisions as well. And getting into the phytochemistry, this is also a very fun part of my research. In this part, I have had the incredible opportunity to collaborate with two undergraduate students studying chemistry under Professor Wenzel at the Claremont Colleges. Together, we extracted 75 samples and we prepared them for running what is called liquid chromatography and mass spectrometry, or LCMS. And this is essentially a method or a way for us to look at all the compounds present in my case, leaf samples. And the way that it works is that the liquid chromatography part, so your sample goes through that column that Ali is holding on the far right, and it separates out the compounds based on their physical properties such as size. And then the mass spectrometry part is what detects the compounds going through the column, and it shows up as these peaks in the spectrum. So this is an example of what a raw spectrum result looks like from an LCMS. This is representing a sample of Eriogonum inflatum, or the desert trumpet buckwheat. And the x-axis is the retention time, or the time it takes for the compound to go through the column. And the y-axis is basically the intensity, or representing the abundance of that compound. And the way that you figure out what you're looking at is by taking into account the retention time as well as the mass to charge ratio. And based off of those two features, you can figure out the compound. And there are many ways to figure this out, but Professor Wenzel and I are collaborating to find an efficient way to analyze this data for all of my samples. So that'll be very cool. And in the meantime, I put this spectrum in a database, and it showed me like what compounds are present. And one of them, two of them I am showing here. And one of those is the leucine, isoleucine DCA. This is an amino acid that that we need as humans, but we cannot produce ourselves. So we need to obtain this through our diets or through supplements. So it's really cool that it was found in Eriogonum inflatum. And I also wanted to highlight this other compound called rutin. And I bring this up because rutin is a very common or very common compound rather, that is being researched in medicine for its effects for helping arthritis symptoms. But another reason I brought up rutin is that it is a flavonoid, or type of phenol, and it is an antioxidant. And this is actually very important for plants that are in extreme environments and habitats, because antioxidants help plants deal with reactive oxygen species that interfere with photosynthesis. So rutin is an example of a compound that a desert adapted plant would have. So that was very exciting to see. As for what's next, so I have finished up all my field work and all of my lab work. So now I'm in the thick of analyzing all my data and trying to make sense of everything that I found out. And with that, I can write my dissertation, which will include a taxonomic treatment of Eriogonum difflexum and relatives, as well as make conservation recommendations. As I mentioned earlier on, there are 5 endemic species that are in the state of Nevada, and many of them are of conservation concern, including the sticky buckwheat, the Darien buckwheat, Eriogonum cancinum, as well as the pagoda buckwheat, which is critically endangered or critically imperiled rather in Nevada, according to NatureServe. So as I get these results and have a better idea of who's who, I can better make informed recommendations for conservation. And last but not least, I need to defend my dissertation, and I'm hoping to finish in the next year or so. And with that, I would like to take the time to thank everyone who has supported me in my research. This goes from grant and funding committees, such as the Nevada Native Plant Society. Thank you so much for making my research possible, as well as collaborators, my advisors, and classmates. Thank you for everyone who has helped make this research possible. And with that, I will take any questions. Thank you.
Thank you very much. That was fabulous. And I bet Jerry's got some questions.
If you have questions, you need to come up here.
Yeah. Yeah, you've got something to say.
Mahima, this is Jerry Tame.
Hi, Jerry. Nice to meet you.
Nice to meet you. We've seen you've missed each other whenever we've been in the same area.
Yeah.
My question is simply, are you going to look at specimens in other herbaria or just stuff at Rancho?
That is a great question. I'm mainly thinking about a lot of the plants I'm looking at are here at Rancho and from my collections, but I've also asked for loans from other herbaria as well, and I hope to look at specimens from other places too.
And just to let you know, ours at Reno are totally imaged, so you can look at them, and if there's a few things you need, let me know. We'd be glad to send them to you.
Yeah, that would be fantastic. Thank you so much.
You're welcome.
Don't think we've got any other questions in the room, but a very nice talk, and thank you for all the teaching you did along the way of things like the phylogenetic trees and things like that. I really appreciated that. Does anyone in the Zoomosphere have any questions or comments?
There's one in the chat, Mahima.
Oh yes, I see the chat now. Let me open it.
Let me see. Could you please share more with rutin? Is it a chemical compound for indications for drought, heat, or cold tolerance? That is a great question. I can speak for the fact that I know that it is found in plants that are growing in drought/heat conditions. I don't know about cold, but I mainly read about rutin, especially found in Cactaceae. But yeah, that's a good question, and I am curious if it is also present in plants dealing with cold as well. Thank you.
Oh, I think that's it. Thank you very, very much. We really appreciate the update on your research, and we look forward to maybe hearing a little bit more when you finish, or following you. We can follow you in different ways to find out what you conclude in reorganizing stuff and things like that. Thank you all who are attending the meeting, both in person and on Zoom, and great. Happy botanizing, everyone, and we'll see you in April. I think it's April 2nd, isn't it? The first Thursday. We look forward to seeing you.